Dear listeners, welcome to another episode of the podcast about enzymes and molecules. I am Roberta Foresti, Professor of Biochemistry at the University of Paris-Escorteil and researcher at the Mondor Institute of Biomedical Research in France. Our new episode is part of a special series that I want to dedicate to interviews with directors of institutes. Being the director of a research institute comes with many challenges, is a lonely job, and the scientists that are given this responsibility often have to learn on the job. For the first episode of this series, I approached Dr Florence Niedergang, director of the Institut Cochin in Paris, who readily agreed to talk to us and share some of her experiences. Dr. Niedergang is an immunologist and obtained a PhD from the University of Paris-Diderot, where she studied the activation of T lymphocytes. She then moved to Lausanne in Switzerland to perform postdoctoral training, working on dendritic cells and macrophages and addressing how these cells interact with bacteria in the gut. After moving back to France, Dr. Niedergang obtained a permanent position at the Centre National de la Recherche Scientifique, CNRS, and first worked at the Curie Institute and then moved to the Institut Cochin in 2005, where she directs her own group, investigating the biology of phagocytes, infection, and immunity. Since 2022, Dr. Niedergang is director of the Institut Cochin. Good morning, Florence. We are very grateful for your time and willingness to talk to us today on the podcast, and I'm really looking forward to the interview with you today.
Good morning, Roberta. I'm glad to discuss with you as well.
Great. Let's start with the first question. Florence, can you tell us briefly about the Institut Cochin? What is it known for? What kind of research is performed there?
Yes, of course. So Institut Cochin is a large biomedical research center. It is located in the heart of Paris, on the same campus as the hospital, so Hôpital Cochin Port-Royal. It is affiliated to different research institutions, national institutions like INSERM, so Medical Research and Health, CNRS, which is leveraging all type of sciences. And we also belong to Université Paris-Cité, that is a large university in Paris. We have more than 600 people working at Institut Cochin, different researchers, clinicians, people who have teaching duties at the university, assistant professors, technical and administrative staff as well, young scientists, PhD candidates, postdoctoral researchers, and interns. We have very intense also training activity with more than 100 PhD candidates and more than 100 interns of various nationalities every year at the Institute. So we all share the same objectives, that is to advance, let's say, knowledge on the biological mechanisms controlling physiological and pathological situations in humans. And we have redefined our main research axes recently that are cancer, immunology, endocrinology and metabolism, microbiology, and what we have called cellular plasticity and reproduction.
I see. Would you say that there is a particular area where the Institute is strong and scientifically, internationally recognized, or do you think there are different areas that are, strengths of the Institute?
Yes. For instance, I mentioned cancer, so it's true that there are some cancers that are maybe more studied here, some solid cancers, some cancers related to the hematology. So we kind of build on the strengths we have and also the relationship we have with the hospital, Cochin. I also mentioned, for instance, endocrinology, metabolism. There is also a large number of teams working on diabetes or other types of metabolic diseases. And this is also in relation with the hospital departments. And of course, I can also mention historically microbiology. There are a lot of groups working on HIV, historically also at the Institute. And the reproduction, I think, is very important. And we want to also further develop this topic at the Institute, because this is also in relation with the largest maternity in France, that is the Port Royal maternity, that is also very close by. And we have also people working together with the clinicians and the maternity.
Yes, and maybe reproduction is not something that many institutes, I think, especially in Paris, are dealing with. So maybe it's a very good idea to
exactly
go in that direction. Yes. So in a way, my second question, which is related to what are the strengths and the weaknesses, you already partially replied to this, but maybe you can go a little bit deeper on this aspect.
Yes, of course. So I mentioned all these axes that we defined. I think the strength is really that we want to work at the interfaces and have people discuss together and really build on this. I mean, the large number of people we have in the Institute, but this is meaning also complementary expertise, the fact that we can, develop ambitious projects based on the combined expertise of the different groups here. So this is really something we want to develop even further. Of course, as I mentioned already, we have bench to bedside projects promoted by the proximity with our colleagues here at the hospital. Then I think we have a very good structuration of the unit in terms of, it's a large unit, but we have organized human resources, financial management, access to the core facilities, very good core facilities, head, engineers who really want to so that we have access to really advanced equipment
support the science,
that support the science, and we try to develop at the same time the projects and the equipment that could be a good opportunity for people to have, and we have also tried recently to take a lot of actions and having a lot of working groups building on the good willing of the people to support the quality of life at work. We have developed what we called groups supporting the career development and discussions with, of course, the engineers, technicians. They had already some support, but this was not so true for postdoctoral fellows and also for researchers who don't always discuss with people, during their career. And we wanted really to improve this. And this has been done for the last 2 years. And we are continuing to develop even further these actions to support career development. I think we have very good attractiveness. We had international calls recently where we could really hire new group leaders. We are really happy to have attracted these excellent new group leaders. And it is really very dynamic building of the unit, with people who come from outside, different perspectives. This is very good for us. And as I mentioned already, we have excellent training activity with more than 100 PhD candidates, meaning around 30 PhD defense per year, which is a lot, and different nationalities also. These are our strengths, I would say. Now for the weaknesses, I think we can still improve our access to European and international grants. There is some possibility to improve there. Then there are 2 other problems that the permanent staff members population is aging. I think this is the case everywhere in France. And while we have success in recruiting researchers based on the national competition, that's very good. But it's more difficult for us to recruit people as engineers or technicians, because this does not depend on national competition. It depends on our supervising bodies. And so it's a bit more difficult to be able to make sure that there is enough people recruited to replace the people who are going to retire. This is a real problem for me for the future. And then another big problem is some of our buildings are in very bad condition. And this is due to the lack of investments over the last years. So this is, for me, a real challenge. I'm trying to convince people to invest and to help us to, to have buildings where we can really do the good science that we want to do. We have a lot of ambition. We have very good people. I need to make sure that we have also, the infrastructure.
Yes.
That fits. And this is not completely in my hands, but I have to fight for that.
Yes. And I suppose this is one part of the job that maybe you were not really prepared for, because you are a scientist and you are not a building company or
exactly,
somebody dealing with this kind of problems. Interesting, because the fact that you mentioned about the aging scientific population, I was reading yesterday that it's basically a problem not just in France but in the US as well, where there are a lot of in the US, in Latin America, many professors don't want to retire, which I understand, or they retire, but they will stay as emeritus. But, there are obviously some positive aspects about that, because they bring a lot of experience, and perhaps even in this kind of semi-retirement mode, they contribute to the life of their university by sitting in committees, or maybe following more the younger generation that needs, like you were saying, advice, career advice, how do you plan for your future, and so on. So but definitely there is a trend of, and maybe it's also related to the fact that there is an aging population in general. People are not retiring, are living longer, and they don't want to retire, they cannot retire so early like they used to.
Yes, you're right. I don't consider this as a problem, because in our case, the persons who are retired cannot be anymore group leaders. These are the rules in the French system. And as you say, they can provide a lot of advice, expertise, and so on. I was mentioning the problem more. For me, and we had success in recruiting young researchers. So this is, for the researchers, is not so much a problem. I really have a problem with the technicians and engineers.
Yes.
Because there is no national competition. We have to ask for positions, and this there is a real shortage, and these are the little fights and discussions with our supervising bodies, so namely the INSERM, CNRS, and the university.
Yes. And perhaps also the landscape in Paris. There are many different institutes where INSERM, CNRS, and the various universities are in a way, have to provide to many different institutes. So we are all competing for the same few positions.
Yes, you're right. And then there is a problem being in Paris of attractiveness, because the salaries are not that high. And of course, it's more difficult to live in Paris than also to live in some other parts of France. So
absolutely. Yes.
So, yeah.
Different
and we need permanent people also. We cannot only live with short-term contracts, because this doesn't provide the good stability for the teams, and you do not transfer the knowledge, the expertise, how to do the protocols, and so on. So I am still convinced that we need permanent people also as engineers. And
I completely agree with you. The loss of lab memories, it's a pain for all of us. Yes. So how do you organize your day? Do you have a routine? Is every day different for you?
So there is no real routine. We try to organize the agenda in advance, so that there is half a day in the lab, half a day for the direction activity. In principle. So then, of course, when I'm attending a scientific meeting, I'm not at the direction. When there is a meeting that is important for direction, like all the units director meeting, then of course I'm not in the lab. So it's not completely fixed, but this is what we try to do, so that I'm sure that there is we really try to split the activities. I also have 2 offices. One for the direction, where basically I discuss a lot with people. People have appointments. We organize this in advance. I deal with all the emergencies. I have appointments with the research unit deputy directors, with the general secretary, and so on and so on. And then I have, half days where I'm in the lab, I discuss with the people there. I'm happy to go back to the lab and to, discuss...
Which life?
Normal life, results, recent results, problems with the protocols, where I try to also sometimes, based on previous experience, give some advice. So this is basically how we try to get organized.
Yeah, that's an interesting organization. I know that some people will say the first 2 days of the week are on one side, but even in this way, it could be that you never lose the plot because you are continuously switching from one role to the other. And therefore, you are not, missing on things that maybe require, your prompt input.
Exactly. And nobody comes to the lab with something to sign, you sign me this letter. Because they know that, in a couple of hours, I will be downstairs signing the letter. So,
exactly.
So this works well, I think, for me, at least, to really have...
So everybody around you is also very clear. Not only it's very clear for you how you organize your day, but the people around you know exactly that they can find you from this time in the director office, and therefore they can count on you. Yeah, this is an interesting way of working. Yes.
And I
would be very efficient.
Yes, and I have a good assistant also who helps me, to get organized and and tells people, okay, this will be signed. Don't worry, it will be signed.
Excellent. So tell me a little bit about yourself. How did you get into science? What's your passion?
Yes. So I think it started when I was young. We had a house in the countryside, and there I was exposed to, looking at little animals, vegetables, growing and so on. And I really loved looking at all the, living organisms that were around me. Then I was attracted by the veterinary studies, medical studies. I thought it was too long. I wanted to be quickly, working. And so I decided to do more, to study biology instead. Finally, I did very long studies, but I had the feeling I was not starting to be engaged for too long, but still I was. But, it was little by little, step by step. And then as a PhD candidate, you have a salary and so on. So it was not exactly the same. And then I think I was really interested in cell communication, the immune system. I studied biochemistry. I could also take full studies for immunology and virology. And I was really interested in all these. I think what we currently do in the lab as type of projects, they really reflect still this interest in combining host pathogen interactions, immune cells, how they work, how they can be hijacked by viruses, especially the phagocytes that are supposed to get rid of bacteria. For instance, when they are infected with viruses, they don't do this properly anymore, so there will be bacterial superinfections. So we combine cell biology, immunology, a little bit of microbiology, and this is something that I still really love, I think.
Yes. So there was no particular lecture or professor or internship that it was really the knowledge that you acquired during your studies that directed you towards the macrophages and this, this field of infection and immunity.
Well, it's as often, I think it started with good teachers for the what we call natural sciences. So all what is, living organisms, geology. This was in high school already. I had very good teachers. I love this. And I love the fact, you ask a question, you try to design the experiment, you get to a conclusion, you redefine the new experiment. And this is this type of game that I still enjoy a lot. And it started, with good teachers. And then little by little, I refined immunology, microbiology, virology. I think we still have projects around these topics that I love. Yeah.
Yes. So if we want to see a little bit more closely what you are doing in your own projects now, in your lab, in your group, what are you working on?
Yes, we are working on. So I started with working on T cells, so T lymphocytes that are very small, round. It was a bit frustrating. It was the beginning of the confocal microscopy, but it was a bit frustrating. So then I really shifted to the phagocytes, so the cells that take up bacteria, debris, present antigens to activate the T cells. So and then I kept this focus on these phagocytic cells. I think they are really fascinating. So the focus is really to understand how they work. They capture debris, they capture bacteria. How do they do this? Then there is degradation. At the same time, the cells get activated. We really want to dissect all this in the, let's say, normal condition. And then try to understand what happens when these cells, instead of, for instance, getting rid of the viruses, they get infected or hijacked by the viruses, and then everything is perturbed. They don't capture bacteria anymore so efficiently, they don't degrade them, they don't get activated so well. So we really try to have the basic knowledge and what happens with the viral infection. And then that leads sometimes to bacterial superinfections because the bacteria can develop instead of being eliminated. So there are these two aspects, and it's really good to have these two at the same time in the lab, because what we develop, let's say for the basic aspect of the research, some types of special microscopy can be used also to study what happens with the virus-infected cells. And at the same time, the viruses are very good cell biologists, so they can, explain sometimes what they actually target in the cell and tell us, okay, see, this is a target that actually is a very important factor in the cell, and we didn't know that before studying the virus.
It was only by looking at how the virus perturbs, like you say, the system, that you then find out some of the targets that are important for the response to this type of infections. Very interesting. How many people do you have in the lab?
So right now we are 14, including interns. This is the high limit, I would say. But there are also, of course, researchers who have projects and they supervise students or PhD candidates. So, of course, there are kind of subgroups in the lab. And we have also, well, depending on the time, different level interns.
Yes. Okay, I think I asked you more or less all the questions, important questions that, I think our listeners will be happy to hear. Thank you so much for your time. It's very interesting to see how people organize their life around these difficult tasks. And I'm very glad to see that you are still very, very interested in science, because I think it's very important that the director of an institute doesn't become too detached from what is the purpose you are there for at the end. You are there to support the science and to implement the future. So maybe you have just one last thing to tell us. What would you really wish would happen? Something very big that could happen in Institut Cochin that you really are working on. In terms of some kind of developments. I don't know if it's drugs or some kind of new discoveries in a very, very important field. Do you have any?
I would like to say that I fully agree with you. For me, it's extremely important to keep this scientific activity, and I'm so happy when I go back to the lab, and and I'm the happiest person if I can go to the microscope and look at cells. So and it helps me, as you say, to keep in touch with the reality and know exactly what the groups need and the problems they are facing, because of course not everything is easy every day. So and the big thing for the Institute, I would say I would like to be able to organize a bit better the research based on organoids and all the 3D tissue, tumoroids and so on, that a lot of groups have started to develop a little bit, but I would like to have this better organized and to support this type of activity at the level of the institute.
Yeah, so
I think it's also the future. We have also to keep in mind that we have to reduce the work with animals. So in line with this, it makes sense to try to develop this activity further. Yes.
So you really want to develop some important technologies. Yes.
Yeah, that will support the scientific questions that people have in the different fields I mentioned before. So it will be useful for cancer, for the reproduction, for a lot of activities. And yeah, if I can help as director to support these scientific projects that people have, yeah, I would be happy to do this in the next couple of years.
Fantastic. I wish you all the success for this.
Thank you very much.
Thank you, Florence. Thank you.
You.
Bye-bye.
Bye-bye.